brain waves, ascending activating system, and sleep promoting centers. Okay? So, sleep is unconsciousness which is reversible by sensory stimulation. Okay? Unconsciousness which is reversible by sensory stimulation. But coma is unconsciousness from which the person cannot be aroused. So this is the difference between coma and sleep. Sleep consumes a third of our lives. So, I don't know how old are you, for example, if you are 30, then you are asleep for 10 years. Okay? And you are awake for 20 years. So a third of your lives usually spend
sleeping
in sleep, right? So it's a considerable time actually.
What is the daily hours of sleeping? In a third, 6 hours, 7 daily hours.
So you mean
What is the estimation of hours, daily hours in the third of our lives? Sleep.
so you mean that the number of hours you need for sleep? 8 or 7 usually. I will talk. We will have this in the Okay. So sleep occurs in all mammals and in all vertebrates. So this shows that during the evolution, sleep was conserved. So this is very important for our physiology. When a characteristic is remained, it means it has some physiological action. It's important for us, for the survival. Okay? So, the clinical importance of the sleep is obvious from the prevalence of the sleep disorders. You know how prevalent are these problems? Insomnia, parasomnia, okay? A lot of things. Sleepwalking, sleep talking, bedwetting, I don't know, nightmares, okay? Sleep paralysis, all of this. So they are very prevalent. So you can find a lot of them in the society. So it shows the importance of this sleep clinic here also. Okay? So sleep need is about 7 to 8 hours for adults, 9 hours for teenager, 17 hours or more for infants. Okay, so you can see here. So at birth, a new birth usually sleeps for 16 hours or more. But there is a decreasing trend in our sleep over time. So when we approach to death, there is an abrupt decrease in the sleep hours a day. Okay, so you can see that here. A newborn sleeps for 16 hours. At 1 year, we sleep for example, 12 hours. At 10, it's about, for example, 9 hours a day. Okay? But in adults, it's about 7 to 8 hours a day. Okay, so sleeping. So as people age, they tend to sleep more lightly and for shorter times at night. So, unintentionally causing embarrassment and loss of productivity and sometimes being hazardous. But it's different from fatigue, which is a feeling of unwillingness or too weak to engage in activities. So when you feel actually don't like to get involved in activities, it means it's a type of fatigue. It's different from sleepiness. Sleepiness is defined by falling asleep unintentionally. Okay? But the causes for excessive daytime sleepiness include insufficient or fragmented sleep during the night. Or sometimes disorders in sleep may happen. Okay? So the reason for excessive daytime sleepiness include insufficient sleep or fragmented sleep. fragmented sleep happens when, for example, a mother has to, for example, feed the newborn, or a person may have prostate, okay, so I mean enlarged prostate, so they have to go to, for example, see every time, okay, so this may imbalance the sleep, so fragmented sleep may happen because of a lot of reasons, right?
During exam also. During exam also.
During exam, maybe. So this is the insufficient sleep during the exam, okay? But there may be some disorders for sleep that cause excessive daytime sleepiness. Why the sleep is not well understood?
Sir, and daytime sleepiness even happen if the fragmented sleep is 8 hours or more.
Yeah, when we have fragmented sleep,
Yeah.
so you may have 8 hours a day for sleep, but the sleep is fragmented, right? So you got enough time, but the quality of the sleep was not normal. So the result will be excessive daytime sleepiness. So in order to be fresh after the sleep, you should have both enough time of the sleep and enough quality of the sleep. Both of them are necessary. Okay? So the reason for having a sleep is maybe energy conservation. These are theories actually. Okay? To replenish glycogen levels in the brain.
So if we are taking sleeping enough, but during daytime,
Yeah.
not in night, is there any physiological changes if you are sleeping daytime, not in night?
So if you have sleep during the day,
But enough sleeping, like not sleeping in the night.
so you mean 8 hours during the day instead of having the,
In the night, yeah.
yeah, actually, there may be some problems, because you are changing the balance of the day and night, So, although you may having the required time of the sleep, but it's happening in the daylight, So actually, it's not maybe proven enough, but there may be some physiological problems. So, night shifting. Okay, night shifting means the same. Okay, so you have to work during the night and you sleep during the day. Usually, they cause some health problem. In this situation. Okay, so physiologists always suggest to sleep during the night, because during the night there is less noise, it's dark, actually less light can reach the retina and the brain, so it can less activate the brain, so the quality of sleep will be better. Okay, so to replenish the brain glycogen level for energy conservation. So this is maybe one reason for having this sleep. Another one is that during the night it's colder, so you should lose more energy for having the body temperature at normal situation, normal condition. Okay, so when you sleep, you spend less energy. Okay, because during the sleep, body temperature is lower. Instead of having 37, you should keep 36. So you will lose less energy when you sleep during the night. But when you sleep during the day, okay, so the body temperature should be kept at 37, so you should lose more energy. Okay, so that's the reason, maybe the second reason for having sleep during the night. And metabolism decreases during the sleep. So all of these may show something about the energy. But sleep is also necessary for survival. It has been shown that if in a rat you prevent sleep, after 1 or 2 weeks, they usually die. Okay? So this shows that also in humans, in order to keep alive, it needs to have enough sleep. Okay? So, in humans, lack of sleep may lead to impaired memory, reduced cognitive ability, and sometimes mood swings, like hallucinations. Okay? So patients with fatal familial insomnia die within several days of onset. So this is a disease that is characterized by hallucination, seizure, loss of motor control, and inability to enter the state of deep sleep. Some believe that their shorter lifespan is attributed to this one, inability to enter the state of deep sleep. Okay, so this is another indication that sleep may be important for survival, for being alive. Okay, so in order to measure the sleep quantitatively, we can use several instruments. For example, we can use EEG, electroencephalography, to measure brain activity. We can use electrooculography, EOG, to measure eye movements. And we can use electromyography, EMG, to measure muscle tone. By these, we can evaluate the sleep. Okay? So quantitative evaluation of the sleep can happen by way of measuring these: electroencephalography, electrooculography, electromyography. So electroencephalography is performed by putting some electrodes on the head. So there are usually 32 of these electrodes. You can put them on the head over the skin.
So,
Okay.
so what do you mean by measuring sleep?
Measuring the sleep, I mean evaluating the sleep. You know,
For
because when a person is sleeping, the only thing that you can see is that they don't move, they close their eyes, only this. But you cannot evaluate the sleep in depth. You don't know what's happening in the brain. Okay? So in order to evaluate the sleep, I mean, any events in the brain during the sleep, you need to record the brain waves, movement of the eyes, or level of activity in the muscles, and also others, for example. So these are some examples, some of the main instruments, but there are also other instruments. For example, measuring the level of oxygen in the blood, measuring the temperature, measuring the flow of the air in and out. Okay, so you should measure the respiration also. So we call this polysomnography. Okay, so polysomnography is a technique for measuring the sleep. Measuring the sleep or evaluating the sleep. Okay? So it has several parts. The main part is EEG. Then we have EOG, EMG, oxygen level, temperature, airflow, okay, heart rate, all of them.
Sir, which part of brain controls sleeping?
So I will talk about this. Okay, let's see one by one. Okay, so electrodes are put on the skin, on the head. They are connected by some wires to a device called amplifier, which amplifies the signals and also filters some unwanted signals. Okay, so the wave is recorded in this way. You can see this on the monitor, or you can print it on paper, right? So when you have ECG, you know ECG, electrocardiography, right? In ECG, you have a special pattern of waves that happens repeatedly. So, example, you have T wave, QRS, right? T wave. It's repeated over time, right? So there is a special pattern in the wave. But here, the waves that are being recorded in the brain doesn't show any pattern. There are only some oscillations. No pattern can be found in these waves. So, instead of having some special waves, the only way that we can use is to measure the number of waves in seconds. So we can rely on frequency only. Okay? So waves that are recorded from the brain are categorized based on their frequency. So we have different frequencies of the waves. The lowest one is this one, delta. Okay? Delta wave has a frequency less than 4 or less than 3.5. So we call them delta waves. So here you can see that in 1 second, you have one wave. So the frequency here is about 1 per second, or 1 hertz. So less than 4 is delta. Then we have theta waves. That's lie between 4 to 7. So if you count the number of waves in 1 second, it should be between 4 and 7. Then you have alpha waves between 8 and 13. And then we will have beta waves, which has a frequency between 14 and 30. Another wave is called gamma, which has a frequency between 31 to 100, or maybe sometimes 200. So we call this gamma waves. So delta waves are the slowest one, the lowest frequency. Then theta, alpha, beta, gamma.
Those waves are determined of what?
Of what?
Yes, some people have delta, some people have alpha. Which changes occur in these two people?
So all of this happen in all people. Okay? So alpha waves are recorded, for example, from the occipital cortex and parietal cortex when the eyes are closed, but the person is awake. Okay? Alpha waves. Beta waves are recorded mainly from the frontal cortex and parietal cortex when the person is awake and the eyes are open, or when the person thinks about something. Gamma waves are recorded. Okay? So when the mind is relaxed, the eyes are closed, alpha wave is recorded. When the eyes are open, beta wave is recorded. When the person thinks about something, so there is something in the brain, right? Gamma waves are recorded. But an adult which is awake and normal usually doesn't show theta waves or delta waves. So it's not normal to record delta waves or theta waves from the brain of an adult during the wakefulness. Okay? But during the sleep it's okay. It's normal to record theta waves or delta waves from an adult brain. But in newborns, it's normal to record delta waves even in wakefulness. In children, it's okay to record theta waves during the wakefulness. Okay? In adults, having theta waves during the wakefulness may show some emotional states like severe disappointment, depression maybe. Okay, so some of these conditions are associated with theta waves during the wakefulness.
So these waves are recorded simultaneously or at different points?
They can be recorded simultaneously from the brain. Okay, for example, so all of this may happen. For example, in some areas of the brain you may have alpha wave, in other way you may have, for example, beta wave. Okay, so it's possible. Or in one point, at this moment, you may have alpha, in another moment you may have beta.
So with different individuals it's different. Like some individuals will have alpha and beta, and in some individuals we have theta and delta. Is it like that?
delta and theta? Yeah, I told you, if you record theta and delta from a person, it means that there was a problem with that person. So normally you should record alpha and beta during the wakefulness. Okay? So here you can see an example. Okay, alpha waves are recorded here. The eyes are closed. The brain is relaxed. The person doesn't think about anything. Alpha is recorded, so we ask the person to open their eyes. By opening the eyes, alpha wave is converted to beta wave. So frequency gets higher, amplitude gets lower. Okay? Then again, the person closes the eyes. You can see that the beta wave is converted again to alpha. So conversion of alpha to beta is called desynchronization. Desynchronization. So only by closing the eyes or opening the eyes you can change your waves, brain waves. Right? Or by getting a deep breath, you can change the brain waves. Okay? When you are stressed out, the brain wave changes. So usually change happens between alpha and beta, but sometimes there may be some beta. Okay? So the waves will show different types of activity in the brain.
Sir, what are these waves? These are action potentials.
No, they are not action potentials. There are some waves called field potentials. So, when you record potentials from one single neuron, right? In order to record this, you should have a very small electrode tip in the scale of micrometer, right? You should put the electrode tip into the neuron. Another electrode should be out of the neuron. So you should measure potential difference between the inside and outside of the neuron. So you are talking about one neuron. So in this neuron you may record resting membrane potential, action potential, synaptic potential, receptor potential. So you have seen all of them. Right? But here, we don't have electrode inside the cell. The cells are in the brain, but you are putting the electrode on the head. Okay? So all of the potentials, usually synaptic potentials, from the neurons beneath the electrode are combined together and reach the electrode. So we call this field potential. Okay? Field means it's a combination of potentials from billions of neurons. Okay? So, this is the field potential, mainly generated by synaptic.
Sir, when these potential changes? When these potential changes?
I told you, in different states of activity. Opening or closing the eyes. Okay? Thinking, disease states, sleep, everything may change them. Okay? So, now, let's see some other important things about the potentials, about the nature of the potential. First, you should consider this neuron. It's a pyramidal neuron in layer 5 of the neuron, of the cortex. Okay? It has dendrites, cell body, and axon. What type of potentials we can record from this neuron? Resting, action, synaptic potential. This is not receptor, so you don't have receptor potential here, right? Because it's a neuron in the cortex. Receptor is in the peripheral. So resting membrane potential, action potential, synaptic potential. So in this neuron you have one axon, right? Action potential goes downward from the brain toward the spinal cord, thalamus. Okay, so action potential is going away from the cortex, away from the electrode. Okay, but Dendrites are closer to the electrode. And you have a lot of synapses on the dendrite. In each dendrite, you will have EPSPs, IPSPs. So at the same time, you may have billions of or millions of EPSPs in a region in the brain. So these EPSPs can combine together and they can generate a very large potential. So this large potential can go beyond the skull and can be recorded by the electrode. Okay, so the type of the potential which is recorded by EEG electrode is field potential. Field potential is generated by synaptic potentials, mainly. Okay, so it shows synaptic activity in the brain. Got it? So now let's see how it happened. For example, you may have some inputs to the cortex, right? For example, these inputs are coming from other areas of the cortex. So, for example, somatosensory area 1 sends some neurons to Broadband area 5 and 7, right? So one area of the cortex is connected with another area. So we have inputs from this area to this area. When you record from the Broadband area 5 and 7, you actually are recording the potential changes that happen as a result of activation in the Broadband area 1, 2, 3, so somatosensory area 1. Okay, so one area of the cortex may activate another. So these fibers are coming from another area of the cortex. When they come, they generate synapses here, release neurotransmitter, neurotransmitters open the cation channels, sodium go into the cell, right? So what happens outside the cell? Positive charges go into the cell. So the outside of the cell becomes negative, right? So you will have positivity inside, negativity outside. Positivity inside goes down this way because it wants to go to here and activate action potential here, right? Meanwhile, I told you before that dendrites are leaky, right? Electric currents leak out of the dendrite. So you will have leak of the current outside. This happens near the soul, right? So you have negativity here, positivity inside, but the reverse pattern happens here. Positive charges are coming out. So outside the cell you will have positivity here. So near the synapse you will have negativity. Away from the synapse you will have positivity. We call this area sink, this area source. So sink is near the synapse, source away from the synapse. Sink is negative, source positive. Right? Did you get this?
Yeah.
So now, imagine that you have some input from thalamus. Instead of having input from cortex, you may have input from thalamus. You should remember from our previous sessions that cortical inputs happen in superficial layers of the cortex. But thalamic inputs come to the layer 4 of the cortex. Do you remember this? I told you that cortex has 6 layers. Layer 4 receives fiber from thalamus. Layer 1 and 2 receive fiber from other cortical regions. So you can see that cortical inputs go to the superficial layers of the cortex. But thalamic inputs come to the layer 4 here. So everything will change here. Sink happens here, source happens here. Did you get this?
No.
You look confused actually. So this is a pyramidal cell in layer 5 of the cortex, right? So cortical input come to layer 1 or 2. Release glutamate. Positive charges go into the cell. You will have positivity here, negativity here. Sorry, sorry again.
Sync.
This is
Negativity
negativity,
and
positivity.
positivity.
So we call this sink, call this source. Then you have cortical input. But thalamocortical input from thalamus. Come to layer 4. So they release glutamate here. You will have sink here. And source here. Positive charges go into the cell. You will have negativity. They go this way and this way. The tip becomes positive, right? So depending on the activity in the cortical input or thalamocortical input, you may have sink and source in the tip or at the base of the endpoint. Always, the location of the synapse is the sink, right? And area 4 is the source. Got it?
Yes.
Sir,
Yes? So, if you have an electrode here, which is recording the potential, right? So, if the cortical input is activated, it will record a negative wave, right? If thalamocortical input is activated, it will record a positive wave, right? So, when you see an EEG, which is like this wave is probably recorded by a mechanism like this. This wave was probably recorded by a mechanism like this. Right? Another thing. If this input releases GABA instead of glutamate, right? What happens? Positive charges like potassium come out of the cell, or chloride go into the cell. The result will be positivity here, negativity here, right? So, in this condition, cortical inputs cause source here, sink here.
They're always opposite, right?
Yeah, always opposite. So, the place that we have negativity is called source. The place that we have sink, the place that we have positivity is source. So, depending on the type of the input and the type of the neurotransmitter, the place of sink and source can be different. If you have sink in superficial layers, you will record negative wave. If you have source near the surface, you will record positive potential. So, these positive and negative waves are generated by activity of inputs and the type of the neurotransmitter which is released.
Sir, these negative charges are in synaptic.
So this negativity, no, this happens outside the cell. Synaptic activity happens inside the cell. So when positive charges go into the synapse, right? Inside the cell becomes positive, but outside becomes negative. So electron is out of the body, it's over the head. So it's recording the actually extracellular space, not intracellular space.
But the synapse is always in the sink, right? Synapse
Synapses are always what?
in the sink.
In the scene?
Sink.
Yeah, if the neurotransmitter is excited. if the neurotransmitter is excitatory, sync is always near the synapse. If the neurotransmitter is inhibitory, sync is away from the synapse. Right?
Sync is for negative outside, right?
Sync is
Negative outside.
negativity, yeah. Negativity outside is called sync. Okay, now, another thing about the EEG. In this image, consider that you have 6 inputs that come to the pyramidal neurons. There are 6 of the pyramidal neurons here, right? Layer 5 of the cortex. So, in first situation, imagine that all of these inputs are activated at the same time. What happens? So neuron 1, 2, 3, 4, 5, 6 receive action potentials at the same time. What happens inside the cell? EPSP. Right? So out of the cell, you will record the opposite. Okay? So negative. Okay. Imagine that these are negativities. Okay? So all of these cells show changes in the potential at the same time. The second action potential arrives, again you will have changes in potential at the same time. The third one arrives, again you will have changes in potential at the same time. Right? So, another condition. Inputs are activated at different times. So each cell may show changes in potential at different times. For example, here the neuron 2 is activated, but this one is inhibited. Neuron 4 is activated at other times. Neuron 4 are activated at different times, right? So all of neurons are activated at different times. So what is recorded by the electrodes, which is located on the head? So the electrode records the sum of these changes, right? So in the condition called synchronized, you can see that all of these changes are summed together. So you will have a large potential in the EEG wave. Then the second change is recorded, the third one like this. But in the irregular activity or asynchronous or non-synchronized, okay, or desynchronized state, you can see that each neuron has different changes in different times. So the sum of these changes are recorded like this. So let's, for example, see this moment, this one. Okay? There is nothing here, nothing here, nothing here, a positive charge here, negative potential here. So they cancel each other. This one is a little larger than this. So you will have a small change in potential here. Some of these are recorded here, right? So at another moment like this, nothing here, nothing here, nothing here, nothing here, there is this one, okay? So there is a small wave here. So at any moment, some of the changes in potential in each cell is recorded by the. So let's compare these two. When you have synchronized activity in the inputs, you will have low frequency wave, which is high in amplitude. When you have irregular activity in the inputs, you will have high frequency wave, which is low in amplitude. It's similar to beta wave. This one is similar to delta wave. Right? So when you record delta wave from the brain, it means neurons in the brain are activated at the same time. Right? When you record beta wave in the EEG, it means neurons below that electrode are activated at different times. Right? Another thing, when you have synchronized activity, potentials are high. When you have desynchronized activity, potential is low. So when frequency increases, amplitude decreases. When frequency decreases, amplitude increases. So delta wave has high potential, beta wave has low. Right? Got it? So, EOG, electrooscillography, is used for recording
Sir, can you repeat?
movements of the eyes.
Can you repeat potential or potential?
Yes,
Right.
here.
Yeah, beta has low frequency.
Low
high
frequency means high potential.
Yeah, when you have low frequency,
high potential, high
potential is
amplitude.
high.
Amplitude you can say.
Amplitude, yeah. Amplitude is voltage here, right? Voltage, amplitude, potential, all refer to the same. So when you have high frequency, amplitude is
Low.
height is low, voltage is low.
This was the example of which fiber?
It's similar to beta wave.
Beta.
This is similar to delta wave, right?
Sir,
So,
like, can you say that field potential is due to the decremental process?
no, decremental process, no. Let's say integration of the EPS, P's and IPs.
Yeah. These are the summation of field potential, right? Sir? 1, 2, 3, 4, 5, 6. Summation of field potential.
Yeah, summation of the field, summation of the field potential.
Yeah.
So, another thing. So let's go on faster, okay? So, in order to record EOG, you can put electrodes here and here. Okay? So, there is always some positive charges in the cornea, okay? And some negative charges behind the retina. So, cornea is always positive, retina is always negative. We call this corneo-retinal standing potential. So, you have two electrodes here, two electrodes here. So this one is active, okay? This one is reference. So, when cornea goes close to the active one, okay? So, this one is active, this one is reference, okay? When cornea goes away from the nose, you will have some negative charges, okay? When it goes close to the lateral electrode, you will have positive charges or positive potential, okay? So, when the eyes go away from the nose and close to the lateral electrode, then you will have positive potential. When the eyes come near to the nose, right? And away from the lateral electrode, then you will have negative potential. So, when you close your eyes, and behind the eyelid, eyes are moving, then cornea can come close to the reference electrode or away from that, okay? So this way you can record changes in potential. So, imagine that the person is asleep, okay? Then the eyes are moving. You cannot see the eyes, but you can record the movement by this thing, by EOG, okay? So we can use this information in order to evaluate different steps of this thing. Based on this, we can say that we have different stages of this thing. Stage 1, 2, 3, 4. Okay, let's see what happens in the EEG in these conditions. When the person is awake, okay, imagine that the person wants to go to sleep, so they go to a room, they turn the lights off, close the door so the noise becomes lower. Okay, so there is no noise. Okay, so the person closes the eyes, the mind is relaxed. So, what should we have in the brain? Which wave? Alpha.
Alpha, yeah.
Eyes are closed, mind is relaxed. So alpha wave is recorded. Before that, there were beta. Okay, so in the awake state, there was beta waves. Then, during the quiet wakefulness, alpha wave is recorded. So now let's see the stage 1 sleep. The person goes into the stage 1. Sleep is started, right? So there will be some waves called spindles. Sleep spindles. So this wave characterizes the stage 1 of the sleep. spindles are usually in the frequency of alpha waves, usually between 12 to 14 hertz or cycle per second. Spindles, okay? So there are alpha waves happening between 12 and 14 hertz. They usually appear for 1 or 2 seconds. So they last 1 or 2 seconds, then they disappear. Okay? After some seconds, they reappear. So, alpha waves, I mean, spindles may show up, right? After that, they're gone, then they regain, they reappear. Okay, so we have repeatedly the appearance of sleep spindles. Anytime they come, they last only for 1 or 2 seconds. When you have seen the first spindle, it means that the sleep was started. Okay, so this is the boundary between the consciousness and unconsciousness. The boundary between wakefulness and sleep. So, what characterizes the sleep? Spindles. Sleep spindles. So this happens in stage 1 sleep, right? Then, in stage 2 and 3, you can record theta waves. I told you, theta waves are not recorded during wakefulness, but they can be recorded during this, right? So in stage 2, you can record theta waves. In stage 3, you can record again theta waves, but there are some delta waves also. Okay, so stage 2 is characterized by theta waves. Stage 3 is characterized by theta waves and some delta waves. Stage 4 is characterized by delta waves. When the dominant wave in the EEG is delta, it means the sleep is in stage 4. Okay? So when more than 50% of waves are delta, we have dominance of delta wave,
Deep sleep.
then it's deep sleep or stage 4. So deep sleep refers to stage 4. So stage 1 to stage 4, you can see that from 1 to 4, there is decreasing the frequency. From alpha to delta. That's what we call these steps slow wave sleep. Sometimes non-REM sleep. Okay, so slow wave sleep means stage 1, 2, 3, 4. Right? So, now let's see this one. This is the time, 8 hours of the night that we sleep. Sorry. Then we have the stages here. So the person is awake for some time, for some minutes. Then the person goes into the stage 1, stay here for some minutes. Then the sleep becomes deeper. The person goes to stage 2 for some other minutes. Then stage 3, stage 4. So from here to here usually takes 30 minutes. Okay? So usually it takes 30 minutes to enter deep sleep. Then the person stays for another 30 minutes in stage 4. Then he or she comes back to stage 3. Then after some minutes they come back to stage 2. Then from here, he or she enters another phase of sleep called REM sleep, R-E-M, which stands for rapid eye movement. Rapid eye movement. So in order to record rapid eye movement, you need EOG. Right? Because you need to know about the movement of the eyes. So rapid eye movement sleep is another phase of this sleep. So in this phase, EEG is like this. Again, beta waves. So beta waves can happen during alert wakefulness or REM sleep. So during the REM sleep, the brain is quite active. In stage 1, 2, 3, 4, the brain becomes actually less and less in activation, right? So frequency of the waves becomes less and less,
Yes.
right? So when you have delta wave, it means neurons are synchronized. So the level of activity decreases. So in the REM sleep, the level of activity is very high because the brain is very active. You can see beta waves. But the person is asleep. It's not conscious. Okay, that's why we call this stage paradoxical sleep. It's a paradox to have beta waves, which is indicator of brain activity, but the person is asleep. It's not unconscious. It's actually the person is unconscious. Okay? So we call this paradoxical sleep. You can ask your question now.
Actually, you answered my question.
Okay, good. So from here, I mean the beginning of the stage 1 to the end of the REM sleep is called one cycle of a sleep. It usually takes 90 minutes. So a cycle of the sleep takes 90 minutes. And you have 5 cycles each night. Okay? Another thing is that you can see that in cycle 1 and cycle 2, you have deep sleep, stage 4. But in the rest of the cycles, you don't have deep sleep, I mean stage 4. So the sleep becomes lighter. So the first cycle is deeper, the last one is lighter. Another thing is that the duration of the REM gradually increases. So the first REM is short, the last REM is longer. So when we go into the sleep, when we go through the sleep becomes lighter and the REM becomes longer. Right? So at the beginning of the sleep, you are very tired. We need the deep sleep because the deep sleep is very restful. But at the end, actually, we are not tired, so we don't need the deep sleep. We more need the REM because REM is important for memory, for learning, for dreaming. Okay, so we need more REM. Okay, so let's compare the REM sleep and the slow wave sleep. During the slow wave sleep, the brain waves are strong. I mean, they have high voltage. And the frequency is low. Right? Delta waves are longer.
So 3 cycles is enough for normal sleeping?
No, 3 cycles is not.
Because at that already there is no deep, what we can say, the stage 4.
Yeah.
Already we are in a level that...
So actually you have rested enough. But the function of the sleep is not only for rest. You need more sleep because you need it for memory, for consolidation of the memory. Okay?
If the timing increase more than 8 hours, so what will happen? So
nothing bad happens, you know? So it's better actually, but 8 hours is enough. Okay, 8 hours is enough for a lot of people. It's like this. For example, for 90% of people maybe. But some people need more than this. Okay, and some people need less. For example, you may have 800 people in your class. Some of you need only 5 hours, maybe. It's enough. Yeah, older people usually sleep less than 8 hours. Yeah. So usually it causes some problem because we need it. Okay? So the light, the deep sleep is necessary for rest. So actually I cannot talk about the exact problems that may happen, but this is not physiology. Okay? Not having a stage 4. I told you before that people that have fatal familial insomnia are unable to go into the deep sleep. Okay, they have only light sleep and they die sooner. Okay, so deep sleep is necessary for us. We may not know at the moment what is the exact mechanism for that. But we know the result, we know the outcome. So let's compare. Slow wave is exceedingly restful. All parameters decrease in these stages. Muscle tone decreases, heart rate decreases, respiratory rate, blood pressure, metabolism, temperature, all of these decrease. But during the REM sleep, which occupies 25% of the total sleep, it's an active form. There is dreaming here. There may be some body movements. It's more difficult to arouse by sensory stimulation in this phase, but more likely to be awakened in this phase. Okay? Muscle tone is exceedingly depressed, so we have atonia. Irregular muscle movements may happen. Heart rate, respiratory rate are irregular. There is increase in the brain metabolism. Remember I told you. Brain waves here are beta. It shows high level of activity. So metabolism in the brain increases 20%. Right? Temperature is lower here. So in this slide, there may be a lot of questions for your final. Okay?
Like, sir, some
We
example.
already did in REM, like randomized.
Actually, dreaming may happen both in REM and non-REM sleep. But REM sleep dreams are longer, more emotional, and more visual, so you can see something in your dream.
But some people we just have a nap and we have a dream.
Yeah, maybe you have this. So especially during the afternoon nap. Okay, so you go very fast to the rest. Okay, so actually I talked about the normal situation during the night. Okay, in most of the people, but sometimes you may go very fast to the rest. And also, there may be dream also in the non-REM sleep. They are shorter, conceptual, but okay, there are differences between them. So, the most important difference between the REM dream and non-REM dream is that the REM dream is vivid. You know vivid? We call this vivid. So, REM dream is vivid. Non-REM dream is dull.
Vivid active.
It means clear.
Clear active.
Like we remember.
Yeah, so we can clearly remember that. But non-REM dream is dull. You only remember something vague. Okay? So these are differences between REM dream and non-REM. Okay? So, again, non-REM sleep, REM sleep. You can see the EOG, electrooculogram. You can see that anytime that the person is in REM, there are activity in the EOG. So there are eye movements. Head movement, I mean EMG in the neck. Okay, head movement is lost during the stage 4. Okay, so there is decrease in the tone of the muscle. Heart rate decreases during the slow wave sleep, and it's very irregular during the REM. Respiratory rate also decreases. Okay, so all parameters usually decrease during the non-REM sleep, and there may be some variations of the parameters in the REM sleep. The questions may be like this: which one decreases more during the REM sleep? Respiration, heart rate, muscle tone, EEG frequency.
Decrease or increase?
Which one increases more during the REM sleep? Respiration, heart rate, muscle tone,
Muscle.
frequency of EEG.
Muscle tone, maybe muscle.
Okay, so when you compare REM and non-REM, okay, heart rate
Increase.
doesn't increase more in the REM than the non-REM. In non-REM you have less, right? Heart rate. The same also applies for respiration. So we have more respiratory rate and heart rate during the REM than non-REM. But tone of the muscle, I told you, tone decreases during non-REM, but is exceedingly depressed during the REM sleep. So we have atonia. So tone of the muscle continue to decrease during the REM. Right? So muscles are very relaxed. So the tone of the muscle decreases more. Temperature also decreases more. But frequency increases. Beta wave, right? Compared to delta wave, right? So questions should be like this actually. For
The
you.
question final like this. The question final.
Okay. So let's ignore this one. So when you have your launch, is it ready now?
No, sir.
Your launch?
No, not
No? Shall we continue
Yes,
or
yes.
we have to get
Sure,
some
sir.
break? Continue?
Continue.
Okay, good. So, okay, here you can see the differences in the dreams or the content of the consciousness. Okay? So during the wakefulness, our thinking process is revealed, so we are aware of our thinking process. We know that what is going on in our brain. Okay? So we call this consciousness. We are conscious, right? And everything is externally generated. So sensation comes from the outside of the body, and we have logical processes in our brain. But in the non-REM state, dreaming is dull and or absent, and thinking processes are logical and perseverating. So thinking process in the non-REM sleep is logical. I mean, the type of the dream that we have here is logical. So everything complies with the wakefulness. Okay, so you have the same rules in the non-REM dream as the wakefulness. Okay, but sensation is vivid in the REM dream, but it's internally generated. And the process of the thinking is illogical, and it's bizarre, so it means strange. So, if you walk during your dream without any leg, right? If you sleep in a desert, okay, so actually it's a bizarre thinking, right?
Irrelevant.
Yeah.
It is
So this is strange, bizarre, and illogical. This happens during the REM dream. But if you did something in your dream, and actually you are ashamed, and you may feel sad for that. So it's logical, right?
Sometimes.
So you are logical as though you did it in the wakefulness. So it's not strange. So it's logical and conceptual. So this usually happens during the non-REM dream.
Like sometimes some people are doing sleep. They change the room. If they don't sleep.
Yeah, this is sleepwalking.
Yeah.
Yeah, it's a problem. So this is a disease. Okay? But now we are talking about dreams, right?
Sir, what
So-
about the one which are speaking during sleep? It is not disease.
Yeah, this is also
This
this.
is disease.
So actually we call this parasomnia. Okay? So parasomnia. Actually, this is not disease. We call this disorder. You know, it's not normal. But if it doesn't your lung, it doesn't harm your lung, it's okay. Okay? But if you speak in a way that actually you get harmed, I mean, something bad may happen for you, then this is the disease, I think. Okay? So sometimes may hit you neighbor or I don't know, anybody else that is speaking with you, okay? So this is the disease actually. So movement is continuous and voluntary during wakefulness, is involuntary during non-REM, but is commanded but inhibited during the REM. So when during the REM dream, you hit something, okay? What does it mean? Your brain generated the motor command for this. Premotor cortex, supplementary cortex planned for that. This plan entered the primary motor cortex. Primary motor cortex generated the command and sent it through the corticospinal tract downward. So this should happen. But it doesn't happen. Why? Because when they come down through the corticospinal tract, when they reach the brainstem, in the brainstem everything is blocked. Okay? So motor command is generated, but it's inhibited by other centers. Okay? If there is any problem in these centers, I mean in this inhibiting center, then everything that you can see in your dream will be acted on. Okay? When you walk, you really walk. When you eat, you really eat. Okay? So motor command is inside the brain during the REM, but it's inhibited. Okay? So again, continuous voluntary during wake, episodic involuntary during non-REM, commanded but inhibited during the. So now let's see what neural mechanisms are responsible for sleep and wakefulness.
Does sleeping posture has any relation with the cycle of the sleep?
Yeah, there are some deviations.
Yes,
Okay. So during the wakefulness, the behavior is like this. So we will have changing movements. Okay, so we can change the position when you are awake. But during the non-REM, the position is the same. During the REM, again, position is the same. So there is no movement. But between the REM and non-REM, the person changed the position. Okay. For example, leg is here, but the nuts is here. Okay. So there was some changes. Okay. Usually it happens between the non-REM and REM. At this, when you switch from non-REM to REM. So let's see the mechanisms for sleep and wakefulness. First of all, let's see an experiment. Okay? If you cut the brainstem here in the midbrain, okay, and record the EEG from the cortex, waves becomes higher in voltage and less in frequency, which is indicative of sleep. Right? During the sleep, we have delta waves, which has high amplitude, low frequency. When you record this from a brain, it means that the brain is sleeping. Right? So when you cut here, result will be sleep. What about cutting here? Between the spinal cord and medulla. Nothing happens. Nothing happens. The waves are low in amplitude, high frequency, beta waves. So cutting the spinal cord, above the spinal cord doesn't cause sleep. Cutting the midbrain causes sleep. Right? So it means there should be here, there should be something here between this cut and this cut in the brain. So neurons should go from here to the cortex, and they keep the cortex active or awake. If you cut these neurons, they cannot reach the cortex goes into the sleep. Actually, this sleep is called actually coma, right? Because it is irreversible. So the same also happens in humans. So this is the cat brain, but here you have human brain. If there is damage here in the midbrain or in the thalamus, the result will be theta waves, which is recorded during the sleep or here the coma state. Okay? So these waves are called K components. Very large waves, that happen in a single pattern, okay? So we call this K complex. So K complex and sleep spindles both happen in the stage 1 of sleep, okay? I told you that the characteristic of the sleep is spindles. But together with the spindles, we may have K complex. Both K complex and sleep spindles originate from the thalamus. Okay? Everything is clear? So far, I told you that there are some neurons in the brainstem, right? They send the axons to the cortex and activate the cortex. The result is wakefulness. If you cut these neurons, consciousness will lose, okay? Then coma is generated or unconsciousness happens, okay? Or if these neurons are inactivated in a physiological process, the outcome will be sleep. So they are responsible for wakefulness. That's why we call them brainstem activating system. Brainstem activating system. They are present in the brainstem, they activate the brain. Okay? Okay. These neurons are located in the reticular formation.
Yes, sir.
So the brainstem is the reticular formation. You know,
Yes.
reticular formation is a stretch from the midbrain to the medulla. We had medullary reticular nuclei, frontal reticular. They were involved in movement.
Like this.
But we have midbrain reticular formation involved in wakefulness. Okay? So, reticular formation of the midbrain and also in the upper part of the pons. Okay? So if you can see the pons into upper part, lower part, the upper part of the pons is also involved in wakefulness. So, reticular formation of the upper pons and midbrain
Midbrain.
is involved in wakefulness. So instead of saying brainstem activating system, now we call them reticular activating system. Reticular activating system. Okay? So this image from the from the Guyton book in the pons. Actually, it's wrong. You know, there are a lot of incorrect things in the Guyton book. It's here, but should be here. Okay, midbrain and upper pons. So there is an excitatory area in the midbrain and upper pons called reticular activating system. Neurons from here send axons to the cortex, they cause wakefulness. That's why we call them reticular activating system. There is an inhibitory area in the lower pons, okay, which inhibits the excitatory area. Okay. Activity in the excitatory area is determined by these. So, peripheral sensory signals. See these signals? They are coming from the spinal cord upward. Pain, touch, pressure, vibration, proprioception, temperature, everything. They are coming from the body. They are going through the brainstem to the cortex, right? Meanwhile, some of them go to the excitatory area. So there are some collaterals from these pathways to the reticular formation. So they activate this area, and this area activates the brain. So when you have pain, when you feel something in your body, you are awake. Okay? They activate the excitatory area, the excitatory activates the brain. Okay? Another thing. There are some feedback signals from the cortex to the excitatory area. So when you think about something, your brain is preoccupied by a thinking process, excitatory area is activated, this area activates the cortex, so there is a positive feedback between these two. Cortex activates ROS activates cortex. So you cannot sleep. Okay? You will be awake actually. So when your brain is occupied by a thinking process with the problem you are thinking about, so sleep is less probable, right? It's difficult to go to sleep. So Another thing that may actually regulate the activity here is the reticular inhibitory network. This one inhibits this and decreases the level of consciousness. Right? So, reticular formation has reticular activating systems located in the midbrain and upper cortex. What type of neurons do we have here? Different types. Excited neurons. Okay, let's see. Some of them are located in the locus ceruleus. They release norepinephrine. Other neurons are located in some nuclei that have gigantocellular neurons. They release acetylcholine. For example, laterodorsal tegmental nucleus, pedunculopontine tegmental nucleus. We'll see them in the next slide. Okay, so they release acetylcholine. Substantial nigra releases dopamine. Raphe nucleus releases serotonin. So, raphe nuclei located in the lower pons are inhibited. Raphe nuclei located in the upper pons and midbrain are excited. So, serotonin neurons may activate the brain or may inhibit the brain. They are generally activated. Okay? So, let's see the human brain. Locus ceruleus release norepinephrine in the cortex. Raphe nuclei release serotonin into the cortex. Laterodorsal tegmental nucleus, pedunculopontine nucleus release acetylcholine in thalamus. The neurons from thalamus go to the cortex. Right? So, not shown here is dopamine neurons. Substantia nigra, ventral tegmental area. Another type of neuron here is glutamate neurons, parabrachial nucleus. Okay? So, norepinephrine neurons in locus ceruleus. Serotonin neurons in raphe magnus, in raphe nuclei. Acetylcholine neurons in laterodorsal tegmental nucleus, pedunculopontine nucleus. Glutamate neurons in parabrachial nucleus. What else? Dopamine neurons in ventral tegmental area and substantia nigra. All of them are excitatory, they activate the brain. We have also histamine neurons here. But histamine neurons are not in the brainstem reticular formation, they are in the hypothalamus. Tuberomammillary nucleus of the hypothalamus. So, some part of the activating system are outside the reticular formation. So, instead of saying reticular activating system, we can say ascending arousal system. Ascending arousal system. So, first we have brainstem activating system. We have seen that neurons are located in the reticular formation. So we call this reticular activating system. Now, we can see some neurons outside the reticular formation. Histamine neurons in the hypothalamus. So we call them arousal. Ascending arousal system. AAS is responsible for wakefulness. What are the neurons? These. Salam. Okay. So, ascending arousal system has these neurons. But there is another group of neurons here in the hypothalamus called ventrolateral preoptic nucleus. Ventrolateral preoptic. VNPO. The neurons here release GABA and GABA. They are inhibitory neurons. These neurons inhibit all of these. So this is the main sleep promoting center in the brain. So all of these are wakefulness centers or activating system. This one is inhibitory system or sleep promoting system. There is another nucleus not shown here, lateral hypothalamus. Okay? Neurons in the lateral hypothalamus release orexin. I try to finish this class in 15 minutes.
It's okay, sir. You can take 30 minutes, but
30 minutes?
don't jugad it.
So, is your lunch ready now?
No, lunch will not come.
Not yet?
You can finish it,
It
sir.
will not send here.
They won't
They
send
will
yet?
not send.
Okay, I thought that you will have your orexin neurons are in the lateral hypothalamus. So, orexin neurons activate all of these. Okay? So,
Activate.
orexin neurons. So, orexin neurons in the lateral hypothalamus activates AAS. VLPO neurons inhibit AAS. VLPO neurons cause sleep. Orexin neurons cause wakefulness.
VLP is also in the hypothalamus, sir. VLPO is also in the hypothalamus.
Yeah, VLPO is in the hypothalamus, puberomammillary nucleus in the hypothalamus, lateral hypothalamus also is in the hypothalamus. Okay? So, let's ignore this one and see these. Okay. This is the firing pattern of activity in the locus coeruleus neuron, which releases norepinephrine. So, if you cut out the locus coeruleus from the brain, cut it out of the brain, and keep it alive and functioning, you can record action potentials like this. So these neurons keep generating action potential even outside the brain. So inside the brain, they are always active. So now, if you count the number of action potential per second in different states, you will see something like this. So here you have wakefulness, slow wave sleep, REM sleep. So during the wakefulness, they are very active. The number of action potential per second is high. When we go into slow wave sleep, they decrease firing. So norepinephrine neurons decrease their firing. They generate fewer action potential. Firing rate decreases. When we enter REM sleep, they stop firing. They don't generate action potentials. When we again get up from the sleep, when we are awake again they increase the fire. So, norepinephrine neurons of the locus coeruleus are highly active during wakefulness, are less active during slow wave sleep, and stop firing or are inactive during REM sleep.
Yes.
The same also happens for histamine neurons, serotonin neurons, dopamine neurons. Let's say monoamine neurons. Okay? So, all of the monoamine neurons in the AAS are active during wakefulness, are less active during slow wave sleep, stop firing, and are inactivated during
REM.
REM. Let's see the acetylcholine neurons. They are highly active during wakefulness, less active during non-REM sleep, and again highly active during the REM. Right, got it? So, now let's summarize these. When monoamine neurons and acetylcholine neurons are highly active, you are awake. Right? When both of these groups are highly active, you are awake. When both of these decrease their firing, we go into the slow waves. Right? When monoamine neurons stop, but acetylcholine neurons increase firing, we go into okay, so this is the mechanism for being awake or being asleep, right? So, if you, so this means that together with activating system, we may have some sleep promoting centers. I mentioned one of them, VLP. But there are also other areas, like these retinuclei in the lower half of the pons. I told you. Nucleus brachis solitorius, suprachiasmatic nucleus, some nuclei in palate. Okay? So, another thing about sleep is that if you prevent an animal from sleep, I mean, if you have sleep deprivation in an animal for some days, then you bring some, then you draw some samples from the CSF of this animal. Okay? You remove or collect some CSF from this animal. Then inject the same CSF into another animal. The second animal sleeps, go to sleep. What does it mean? It means during the sleep deprivation, there are build-up of some chemicals in the brain. These chemicals are sleep promoting, so we call them somnogens. When you inject them into another animal, they cause sleep in the second animal. Okay? So during wakefulness, there are accumulation or build-up of some chemicals which are somnogens. The names of these chemicals are these: urabin peptide, another one is, so it's not here, delta sleep inducing factor. Delta sleep inducing factor. Another one is adenosine. Interleukin 1B. Okay, this is the main one, adenosine. We have also interleukin 1B, delta sleep inducing factor, and urabin. So they increase in concentration in the CSF.
So sleeping pills are adenosine like this?
Sorry?
Sleeping pills.
Yeah.
When some is taking, so.
Actually, these pills usually use other mechanisms like activating melatonin. You know, because melatonin also have some functions on these neurons. Okay? So there are melatonin compounds usually. But if you activate adenosine receptors, you can cause a sleep. Okay? Because it's a very powerful somnogen, adenosine. So, what happens if you remove these nuclei? Nuclei that cause sleep. Okay? So, usually the animal dies because of the tiredness, but the animal cannot go to sleep. Okay? So, we have AAS, ascending arousal system, sleep promoting centers. If you remove sleep promoting centers, the AAS is active, so causes intense wakefulness, which is very prolonged. It causes tiredness in the person or in the animal. The person or the animal cannot go into this sleep. Okay? They never go to sleep. So they die out of the tiredness, but they cannot sleep. Okay? What does it mean? It means in order to go to sleep, in order to fall asleep, you need sleep promoting center. The tiredness itself is not enough. Okay? In the past, we thought that, okay, we are awake, okay, and we do some mental activity, then we get tired, and after that we sleep. So tiredness causes loss of activity in neurons, and neurons become inactive, so we go to sleep. Okay? This was a past view. But now, we know that tiredness is not enough. Although the neurons are tired, but they cannot go to sleep. In order to go to sleep, you need sleep promoting centers. So sleep is an active activity, right? It's not a passive activity. It's active activity. So some brain areas should be active in order to go to sleep. So sleep doesn't mean lowering the activity of the brain totally. Some areas actually show decreased activity, but some other areas show
High activity.
higher activity during this sleep, right? So, cholinergic nuclei, locus coeruleus nucleus, wakeful nucleus, tuberomobiliar nucleus. This one releases acetylcholine, norepinephrine, serotonin, histamine. They cause wakefulness. Orexin activates all of them. VLBO inhibits all of them. Orexin neurons cause wakefulness because they activate these. VLBO neurons cause sleep because all of these are inhibited. So, this nucleus, the blue one, is VLBO. The red one is lateral hypothalamus. This one releases orexin, this one releases GABA. So these two nuclei in the hypothalamus determine if you are awake or okay? So when the level of activity in the orexin neuron is higher than the VLBO, we will be awake because all of these neurons are active.
Active.
When the VLBO neurons are more active than the orexin neuron, then all of these are inhibited and we will go to sleep. Got it?
Yes.
So I will finish my discussion here by a disease. We're talking about a disease called narcolepsy. Let's see the signs and the problems that the patient has. Okay. Excessive daytime sleepiness. Remember? I told you at the beginning of this session. Excessive sleepiness, excessive daytime sleepiness, and irresistible sleep attacks. So the patient feels sleepy and sometimes unintentionally fall asleep, right? So there are sleep attacks that usually last for 20 minutes, which is precipitated by passive and boring behavior, like physiological aspect. Yeah. So, the second consequence in this disease is cataplegia, which is a sudden bilateral loss of the muscle tone. So the person suddenly loses all the muscle tone, so they fall down, but the person is conscious. Okay? So the person feels that something wrong is happening, and he or she knows that actually they fall down, but they are awake. Okay, so we call this cataplegia. The person is conscious. It happens only for some seconds. Usually it's intensified by laughter. So when the person laughs, they fall, actually, because they lose their muscle tone. And another consequence is hypnagogic, hypnopompic, dreamlike hallucination. So at the beginning of this sleep, or at the end of this sleep, when there is transition between wakefulness and sleep, okay, the person feels some hallucinations. So it's like dream, but the person is aware that they are not dreaming. They are hallucinations, okay? The images are not real. Okay, so we call this dreamlike hallucinations. Sleep paralysis also happens. Again, the transition between the sleep and wakefulness. So the person feels all of the muscles are actually relaxed. There is atonia. The person cannot move any part of the body. Okay? Again, the patient is awake, and he or she is conscious, and she knows that what's happening now. Okay? So it lasts longer. The person is unable to make even the smallest movement. And so this is the mechanism, I don't know, okay? There is also disturbed nocturnal sleep. So the sleep is short, and the latency to go to sleep is very short, so the person sleeps very fast and usually begins with the REM sleep. So instead of having a stage 1, 2, 3, 4, 3, 2 REM, the patient goes directly to REM. Okay? So symptoms of excessive daytime sleepiness and sleep attacks. So the second one, cataplexic, loss of the muscle tone, dreamlike hallucinations, sleep paralysis, and disturbed nocturnal sleep. So if two of these are present, usually we say that the patient has narcolepsy. Okay? Or if these three are present, again, we will say that the patient has narcolepsy. So in the past, we didn't know the reason for having narcolepsy. But now, scientists discovered that there is a problem with orexin neurons. Okay? So you can see a special type of scaling of the neurons called immunohistochemistry. Okay? By this type of scaling, you can use some antibodies in order to scale some neurons. So here, antibody against orexin was used. So orexin neurons were scaled. So these black dots are orexin neurons. This is the fornix, lateral hypothalamus. Okay? So you know that lateral hypothalamus is located lateral to the fornix, and medial hypothalamus located medial to the fornix. So you can see that in the lateral hypothalamus and perifornical area, you will have a lot of orexin neurons. So this is the brain of a normal person. But after the death of the narcoleptic person, they stain the brain with the same method. There is no orexin neuron here. So loss of orexin neurons cause narcolepsy. Okay? Orexin neurons activate all other neurons in the AAS. They cause wakefulness. Then they are lost. AAS cannot be activated enough. So there is some sleep attacks, daytime sleepiness, I don't know, epilepsy and other signs. Okay? So that's all about the sleep. We have reached this. So let's have a lunch time.